This is Unit 2, Part 4. We're going to discuss active transport mechanisms. Before, in Unit 4, Part 1, we spoke about passive transport mechanisms at the end. And here we'll start talking about active transport mechanisms, which all require energy. And typically the form of energy input, the source of energy is from ATP that the cell has built up as a storage form of energy that's readily accessible to it. These require these systems require carrier proteins as part of the transport mechanism and the use of energy. So there are some primary active transport and secondary active transport mechanisms, but I'm not going to go through that. I'm just going to speak more generally about active transport and talk about particularly mostly about one type of active transport mechanisms involving the movement of sodium and potassium across the cell membrane. This mechanism of active transport is called the sodium-potassium pump and is present in all cells, and is a way in which cells move sodium out of the cell and potassium in. Please don't get confused by this diagram in the middle here, which is showing the sodium moving in and the potassium moving out. The sodium-potassium pump pumps sodium out and potassium in. What's shown here in the middle is that in terms of concentration gradient, potassium is higher inside the cell and sodium is higher outside of the cell. Potassium is higher inside than outside, and sodium is higher outside than inside, due to the action of the sodium-potassium pump. So if there are channels, specific channels, either a sodium channel or a potassium channel that opens, then you'll get a passive movement or passive transport of sodium in and potassium out, as is shown here in the middle. They will move sodium in and potassium out because of the concentration gradient. So potassium gets pushed out because it's higher inside than outside, and sodium would move in because through a channel because it's higher outside than in. So that's the situation in terms of passive transportation and concentration gradients. However, all of that does occur, or that situation is present because of the action of the sodium-potassium pump. Now, the sodium-potassium pump involves a particular protein molecule embedded, as shown here in purple, embedded in the cell membrane, and it has the ability to move sodium out. When sodium moves into association with this protein, then ATP causes phosphorylation of the protein. So what you get is a phosphate group from ATP binding onto the protein molecule, so it's now phosphorylated. And that causes a change in shape. Remember, structure subserves function. So it has a particular function now because now it's taken on a new shape, and that function is it now can pump sodium out of the cell. Okay? The energy input is from the ATP. Now that it's phosphorylated, it has the ability to pump the sodium out. That also ends up allowing it now to bind extracellular potassium, and that potassium moves in as the phosphate molecule gets released here. So this is a process that is continually happening in all cells and is the reason why cells have a negative charge on the inside. That is called, the fact that they have a negative charge relative to the outside, that's a separation of charges, and that's called a membrane potential. So cells have a membrane. I'll write it here on the side, although it's not easy to write with this. Membrane potential. So all cells have this membrane potential. The potential is a separation of charges, and it's a negative charge inside the cell versus more positive charge outside. So it's negative inside relative to the outside. That, although it's a little difficult to see, is an O, and this is an E. So that's membrane potential. The fact that 3 positive sodiums move out and 2 positive potassiums move in causes a relative shift in charge so that the inside is negative relative to the outside. That's quite important in all tissues, but it's particularly important in 2 types of what are called excitable tissue. Now, you have 2 major kinds of excitable tissue in your body. One is nerve tissue, and the other one is muscle tissue. And these 2 types of tissue use that membrane potential because if they get the right signal, it can trigger a sudden opening of channels. Now remember, as shown in the middle of this diagram, because of the concentration gradient differences set up by the sodium-potassium pump, you have the possibility of this, if a channel is open, a sodium channel or a potassium channel, then you have a potential sudden rush in of sodium or out of potassium, right? Okay, let's move on. Now, when you're talking about direction of movement, you can also characterize active transport mechanisms according to which direction the substances move. If they both move, if more than one substance is moved through a transport mechanism and they're both moving in the same direction, then the active transport mechanism is called a symport system. If the 2 are moving in opposite directions, then it's an antiport system. And as you can see, because with the sodium-potassium pump, sodium is going out, is being moved out, and potassium is being moved in, and this is a antiport system. With the case of sodium and glucose moving in, this is a carrier protein for this kind of transport of glucose along with sodium, and they both move in, so this is a symport mechanism of moving the 2 into the cell. Now, a further classification of transport is not just by movement of individual types of ions or charged particles or individual molecules, but involves a larger structure called a vesicle. This is also an active transport mechanism. All of these types of vesicular transport require ATP, so all of them are active transport mechanisms. Vesicular transport involves the movement of either molecules in solution in fluid, solubilized in fluid, or solid molecules, solid structures that are surrounded by a phospholipid bilayer vesicle. And they can either, that's a vesicular transport mechanism, a subcategory of active transport, and it can be either out of the cell, movement out and via vesicular transport out of the cell, which in which case it's called exocytosis, or it can be movement into the cell, in which case it's called endocytosis. So we have passive transport not requiring energy, active transport requiring energy. We have transport of individual molecules across the semipermeable membrane in an active transport mechanism that involves input of energy, or we have movement of molecules inside of membrane-bound vesicles, which is a vesicular transport, which is a form of active transport. And if it goes out, it's called exocytosis, and if it moves in, it's called endocytosis. Furthermore, we have subcategories of vesicular transport, endocytosis, involving vesicles. And there are a number of mechanisms described here. I will not go into each of them, but I will talk about phagocytosis and pinocytosis. So, as we see here on the next slide, in terms of vesicular transport, as I told you before, it can be transport of large solid particles, macromolecules, or fluid, and it can involve movement out of the membrane, or out of the cell, or into the cell. And all of them, if it's a vesicular transport, it involves whatever is being transported is surrounded inside a membrane-bound vesicle. So a phospholipid membrane-bound vesicle. Here you see an example of exocytosis. Here there are some molecules that the cell needs to secrete. So the cell has synthesized these molecules and will secrete it. There's lots of secretory types of cells in the body. You see the phospholipid bilayer surrounding the molecules inside this vesicle, and the vesicle moves up to the plasma membrane, the cell membrane, and fuses it. Very similar in many ways to how, although a little more complicated structure, certainly, similar to a bubble moving and coming into contact with a larger bubble and then just fusing with it. So the phospholipid membrane of the vesicle actually becomes part of the plasma membrane, and therefore the contents inside, the molecules that were inside, move out are now continuous with the outside and are secreted out in this way. That's a good example of exocytosis, vesicle transport, and active transport mechanism. There are other types of ways of looking at vesicular transport in terms of where is the vesicle moving. If it moves into a cell, across the cell, and then out, if it's being transported, sometimes cells are arranged in layers. So if it comes in from the bottom, moves through the cell, and then is secreted out the top of the cell into a different area of the body, that type of transport is called transcytosis, where the molecules are inside a vesicle, so it's vesicular, and they're moving in, across, and out of a cell. If the transport, another subcategory of way of looking at vesicular transport is that if it's just movement within the cell itself, that's called vesicular trafficking, just moving of substances that are bound inside a vesicle from one area of the cell to another area of the cell. Well, I told you there were a number of different mechanisms of endocytosis, vesicular transport that are endocytosis, involving movement of either solids or fluids into a cell. Movement of solids into the cell is called phagocytosis. Movement of liquids into a cell is called pinocytosis. So it also is called fluid phase endocytosis. So pinocytosis means that the plasma membrane folds around an area of extracellular fluid and draws it in, forming a vesicle on the inside. Phagocytosis, the membrane stretches out around a solid object outside of the cell and then brings it into the cell inside of a membrane-bound vesicle. Here you see a mechanism that I will not talk about, so let's move on. No, let's move on. I don't want to talk about this kind of, well, it does show one thing. It shows that this vesicle which is formed is moving into the cell and across the cell and then out on the other side. So that is a type of transcytosis. This is, I'm not going to talk about the one on the left, but this is an example of phagocytosis, another type, subcategory of endocytosis, where the ingested particle is solid and the cell membrane forms what are called pseudopods. So they stretch out around the solid object. Here's one pseudopod on one side. All of this is a pseudopod. Here's a pseudopod on the other side, stretching out. They stretched out and surrounded the solid object, and eventually will actually fuse at this area right here, so that you get, in the end, a vesicle, membrane-bound vesicle with the solid object inside of it. That is called phagocytosis. There are two major types of cells in the body. The major cells that mediate phagocytosis in the body are called macrophages, and the other one is neutrophils. There are other cells that can carry this out, but most phagocytosis is mediated by either macrophages or neutrophils. It's quite difficult writing with this neutrophil. All right. This is a table that lists all different kinds of active membrane transport mechanisms. Energy source, obviously, if it's active transport, the energy source is always ATP. All these, they're active, so they all require energy. Here are some examples. I haven't gone through all of these, but there is active transport of solutes. That's movement of ions, and I talked about that with the sodium-potassium pump, for example. If it's movement out, then it's exocytosis. If it's movement in, it's endocytosis. And I talked about two examples of endocytosis surrounded by vesicular transport. Examples of vesicular transport endocytosis. One was phagocytosis and the other one was pinocytosis. Okay, now I want to remind you that I did mention when we were talking about the sodium-potassium pump that the fact that there is a separation of charge across the membrane due to the action of the sodium-potassium pump, continually pumping sodium out and potassium in. 3 sodiums go out, 2 potassiums come in. So you end up with a negative charge, and that separation of charge or voltage across the membrane is called the membrane potential. And normally it's stable, so it's a resting membrane potential. And the internal charge is negative and can vary depending upon the cell, anywhere from -20 to -200 millivolts on the inside. Again, it's because of this separation of charges and the different concentration gradients and the relative more sodium that was moved out and potassium that was moved in. So that membrane potential is in a steady state, and it's maintained by the active transport of ions by the sodium-potassium pump. And here you see a diagram showing the cell membrane, and the sodium-potassium pump is shown here. No, I'm sorry, I'll take that back. Let me just erase it. Yeah, that doesn't show that. What you have here is an example of a channel. Remember, the channel is not the same as a pump. Channels are specific for particular ions. Here we are looking at a potassium channel. It will allow potassium across, and it's a case of passive transport here, where it doesn't require energy, and the movement in terms of the channel is due to a higher concentration of potassium inside versus outside. So potassium, if the channel is open, then potassium will move out. And I can tell you that potassium can also move the other way. So it's in balance, but mostly it's leaking out because of the action of the sodium-potassium pump, which is building up a higher concentration of potassium inside as opposed to outside. So here we have potassium leaking out through the channels. These potassium channels in many cells are open. They're not particularly open to a great extent. They leak potassium out because potassium, after all, is a higher concentration inside than outside. But there is a certain limit to how much potassium will leak out, because at the same time, potassium does move into the cell due to attraction of the positive charge for the internal negative charge that's present inside the cell. Okay, let's move on to a completely different topic, away from transport. In this case, you should understand that there are molecules on the surface of cells on the outside that are called cell adhesion molecules that allow cells to interact with other kinds of molecules. So, and they help anchor cells to other structures. They assist cells in movement across, and sometimes in binding or moving cells into an area of injury, like the movement of white blood cells to injured or infected areas. Involves these cell adhesion molecules. In the end, in a multicellular organism, you need interaction between the different cells in different parts of the body, and that can occur because of particular molecules protein molecules, or sometimes glycoprotein or glycolipid or protein molecules that they express at their cell surface, right? So they'll be able to interact if there's a specific protein that can respond to something in the extracellular environment, which was produced by secretion from some other cell. So contact between cells due to receptors and interaction because of receptor interaction is important in normal development and communication and normal physiological function and normal immunity, because you always have to be able to recognize self from non-self. So those membrane molecules or receptors can, designate something as being self for the immune system. You also have molecules on the surface which can act as gated channels. I'll explain. You'll learn about gates later. But they respond to changes in charge. So a change in the membrane potential can trigger gates to open. And this is particularly important in excitable tissues like nerve and muscle tissue. Sometimes you have chemical signaling where a soluble molecule will bind to a specific receptor molecule. Neurotransmitters are efficient at binding to specific receptors in nerve and muscle and triggering these gates to open. If it's responding to a biochemical molecule like a neurotransmitter, then it's a chemically gated channel. It can bind its neurotransmitter, and then it responds by opening or closing a gate, okay, on the channel. And then there are what are called G-linked receptors. I'm not going to get into that. Suffice it to say, you have all kinds of molecules on the surface of cells, and depending on what cell we're talking about, it could have a different set of molecules expressed on its surface from another cell type. So, white blood cell will have different molecules expressed on its surface from a muscle cell, and they're all different from a nerve cell. So cells become specialized during development. They differentiate towards a specialized form. They start expressing on their cell surface within the membrane different proteins that can act as sometimes channels, sometimes pumps, sometimes receptors for a certain signal molecule. And in this way, the cell interacts with its environment within the context of the entire body. A multicellular organism needs to be able to send signals around the body and interact from cell to cell. And this is how it does it, at the surface through the membrane typically. That's the end of Unit 2, Part 4.